Hello everyone, Science Spot is a podcast that shines a spotlight on the latest scientific technology from Japan. Your host is Ryan from Science Talk. So, when creatures move from the surface to underground environments, especially dark places like caves, they often undergo remarkable evolutionary changes. For instance, as you might know, fish and insects living in caves frequently show degradation of their eyes or a reduction in body pigment. This happens because in the absence of light, eyes become unnecessary and body coloration no longer serves a protective purpose. It's almost as if, like unused muscles, the body adapts and changes in response to its environment. With recent advancement in genomic analysis, it's becoming clear that this morphological degradation is closely linked to the degradation of the genes responsible for those structures. Underground organisms are excellent subjects for studying genome evolution because the connection between environmental factors and the gene degradation is so clear. What's even more interesting is that some closely related underground species have different evolutionary origins for their subterranean colonization. By studying such groups, researchers can explore whether gene degradation is merely a random accumulation of changes, or if it exhibits a certain directionality when adapting to similar environments. The focus of this study was on a group of small insects called tracing beetles, which are only a few millimeters to about 1 centimeter long. Over 400 species are known in Japan, including lineage that are believed to have independently colonized caves, just like the one studied here. The research team chose these tracing beetles to investigate if there are rules governing the degradation of vision-related genes, which are linked to their eye degradation. This research was conducted by a team including a PhD student at Hokkaido University and at the time and is now a researcher at Kyoto University. And they collected a total of 4 Thracian beetle species from various locations across Japan. Specifically, they sampled 2 cave-dwelling species with completely lost compound eyes. The 4 types of this beetle, and in the lab, they extracted DNA and RNA from the sample and used state-of-the-art sequencer to obtain gene and gene expression information. With this data, they estimated the evolutionary relationships among these beetles and specifically focused on analyzing 24 vision-related genes found in insects. A gene was considered retained if its amino acid coding region was conserved. Conversely, a gene was deemed lost if mutations causing loss of function were detected, or if the gene itself could not be found within the genomic information. First, they confirmed the 2 cave-dwelling tracing beetle species in their study belonged to different evolutionary lineages. This finding aligns with previous morphological studies indicating that these 2 species did not evolve from a common cave-dwelling ancestor but independently colonized caves. Yet both lost their eyes. This suggests that their similar eye degradation is a result of parallel evolution due to the common selective pressure of living in darkness. Next, when they examined the 24 vision-related genes, they found striking results, and Despite their independent cave colonization, 9 genes were commonly lost in both cave species, and 12 genes were commonly retained. The commonly lost genes included those for light reception, such as opsin genes, as well as IRR and TLP genes, involved in the signal transduction in photoreceptor cells, and cardinal genes involved in pigment synthesis. This indicated a high consistency in the evolutionary fate of vision-related genes and in these two trace in vitro species, despite their separate evolutionary path to cave life. Furthermore, the research team investigated what characteristics the commonly lost genes shared. They focused on the gene multifunctionality or pleiotropy. Multifunctionality referred to how many different types of tissues a single gene functions in. They assessed this using gene expression that from Drosophila melanogaster as a reference. The results show that the commonly lost genes tend to have low multifunctionality, and this means they were typically expressed in only a limited number of tissues. Conversely, the commonly retained genes tend to have high multifunctionality, being expressed in multiple tissues. It suggests that even in a cave environment where eyes are no longer indeed needed, and not all vision-related genes degraded randomly. This research highlights that the degradation of vision-related genes in cave-dwelling tracing beetles is not simply a random accumulation of changes, but has a certain directionality. And one key factor determining this directionality appears to be the gene's inherent property of multifunctionality. This discovery could pave the way for predicting which genes are more likely to evolve or degrade across various organisms based on their genomic information. This wholly significant promise, potentially even offering clues for assessing disease risk and prevention in the human genome. That's all for today's Science Spot. This podcast is broadcast daily on workday morning in both Japanese and English. Honestly, I'm absent because I went to Japan and Shanghai in China. I took a rest, but now I restarted this broadcasting. I love for you to listen to the podcast and post your notes and thoughts with the hashtag ScienceBot. See you next time.